Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Maria Gambetta from the University of Lausanne on this show. Please let me briefly introduce you to our audience. You got your PhD in 2010 from the EMBL in Heidelberg. After that, you moved on to the postdoc at the Max Planck Institute for Biochemistry in Martinsried and at the EMBL in Heidelberg. After that, in 2018, you moved to the Center for Integrative Genomics at the University of Lausanne as a tenure-track assistant professor, and you are still there today. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
So my mother was an interpreter for the United Nations, and my father was a civil engineer and working in South America. And so I grew up in New York in a very multicultural environment. I was first in the United Nations school. And working in a multicultural environment was something that is since then very appealing to me. So I very much enjoy working in an endeavor that transcends borders. That's one aspect of it. The other aspect is that I have always loved to think creatively about things, and I've always hated to memorize things. And so studying science was a way to think about problems in a creative way. And in particular, I was always attracted to molecular biology, which is more abstract than studying macroscopic things, right? Such as, plants or ecosystems. And finally, there's another aspect about working and being a scientist that really fits my personality, which is that there's something nonconformist about being a scientist, in the sense that you're studying things that are not made by humans, right? They don't obey the rules of human society or human nature, and so studying something that goes beyond that was always attractive to me.
Yeah, let's talk about your science that centers around the molecular basis of gene regulation, specificity in Drosophila using genetics, genomics, biochemistry, and also live imaging tools, which you just described as not so appealing to you. Let's see how this fits into that. So let's start in the year 2009. There you were first author on a science paper where you investigated the essential role of the glycosyltransferase SXC-OGT in polycomb repression. Can you talk about this story?
Right. So I did my PhD in Jörg Müller's lab, and I got a cool project, which was to characterize a polycomb mutant. So polycomb proteins. There are about 20 of them in Drosophila, and mutation of any one of them leads to loss of repression of a common set of target genes. And there was one of these mutants. It was called SXC for super sex combs. In Drosophila, many polycomb mutants have the name sex combs in them, right? Like extra sex combs, super sex combs, posterior sex combs, blah, blah, blah. This is due to the fact that these mutants all have homeotic transformations because they misexpress Hox genes. And one of these homeotic transformations is the appearance of sex combs, which are, dark hairs that are normally only present on the first pair of legs. In males, actually, all pairs of legs get them, right? Because the Hox gene that is responsible for forming these sex combs is inappropriately expressed in all legs instead of only in the first pair. Anyway, that was a parenthesis. So right, the super sex combs mutation, no one knew which gene had been mutated and was responsible for that phenotype. And so I embarked on this project to try to figure out which gene had been mutated and after 1.5 years, I finally found mutations in the O-glucnec transferase gene. This is a conserved glycosyl transferase that's present from flies to humans. And it was the only known glycosyl transferase that's actually present in the nucleus and in the cytosol of cells, right? Whereas most glycosyl transferases are in the secretory pathway, right? In the endoplasmic reticulum and in the Golgi, because they glycosylate proteins that will then be secreted or exposed at the plasma membrane at the cell surface. So anyway, so OGT, what it does is that it adds single glucnec, which is N-acetylglucosamine residues, onto serins or threonines of a plethora of proteins. Okay, and so there had been hundreds of proteins that had been described to be glucnec modified, and yet in Drosophila, if you mutate OGT, you get a remarkably specific polycomb phenotype, okay, with misexpression of Hox genes and everything. And so this paper reported this discovery, and we had also profiled glucnec for the first time on chromatin by ChIP-seq at that time, right? And we had seen that glucnec peaks are present at sites where polycomb proteins bind. So in Drosophila, polycomb proteins bind at elements called polycomb response elements, or PREs, and that's where we saw these peaks appearing. And finally, another important piece of data in that paper was that I found that one specific other polycomb protein that's called polyhomeotic, it was specifically glucnec modified. So of all of the polycomb proteins that I tested, and I tested about 10 of them because we had a large collection of antibodies, I found that only polyhomeotic was glucnec modified by OGT. Mm-hmm. So that was that. Yeah.
So you then were part of a follow-up work where you reported the crystal structure of the core of the Drosophila PCG protein complex, Faux repressive complex. So could you introduce us to this complex? And I mean, it's always nice to have a crystal structure, but I'm always a little bit intrigued by what exactly does it, what kind of level of information does it bring when you have the crystal structure? So what did it do for you?
Yeah, right. So I must say, I was co-first author on that study. Really, the first author, it was Claudio Alfieri, who was a PhD student in Christoph Müller's lab, a structural biology lab at EMBL. And so Claudio had crystallized the interaction interface between Faux, pleiohomeotic. It was the first known polycomb protein with a DNA binding domain that can recognize a specific sequence that is enriched in these PRE elements that I mentioned before, which in Drosophila correspond to the sites where polycomb is targeted mainly. And so he had crystallized the interaction interface between a fragment in Faux and a fragment in its interaction partner called SFMBT, which is another polycomb protein. And so this crystal structure gave us atomic resolution of the interaction interface between these proteins. And this allowed Claudio to design point mutations that killed the interaction between Faux and SFMBT. My contribution to that study was to then describe the biological consequences of these point mutations. So what happens in flies? And basically, well, as you would predict, point mutating these residues strongly decreased the interaction between Faux and SFMBT. This led to impaired targeting of SFMBT to chromatin, and this resulted in de-repression of Hox genes. And so this was, the message of this study was that basically the direct interaction between Faux, this DNA targeting polycomb protein, and SFMBT, which is which is a partner protein that can interact with members of PRC1, for example, another polycomb repressive complex, that this was important for polycomb function. Yeah.
So this was work that is maybe not so related to your current work. So you then switched to the lab or moved to the lab of Eileen Furlong, and there you switched to investigating CTCF function in developmental progression. And this is maybe more the work you're focusing on right now. So maybe you can give us a little bit of insight into the first work in this area that you did in Eileen Furlong's lab.
Right. So my main project in Eileen's lab was actually not related to CTCF. CTCF was a side project that I started on the side. Yeah, that's the one that was published first. But let's say the heart of my work in Eileen's lab was to investigate whether 3D contacts between enhancers and promoters are dynamic, okay, over time, for example, over embryonic progression and across cell types. Because if that were the case, it would suggest that that modulating the physical proximity between enhancers and promoters could be a strategic way to regulate gene expression, right?
Sorry to interrupt. In a former episode, like just a couple of episodes ago, we talked about like the distance. So the distance on the primary sequence of the DNA, what kind of impact this has on like enhancer promoter interactions. But here it was like more a timely thing, right? So not like the distance, but more in the third dimension then.
Yeah. So it was we were basically comparing different cell types, neurons and muscle, and different time points in embryogenesis. And looking, so we did this using two approaches. One is chromosome conformation capture, and we were using Capture-C technology that was, using a protocol that was generously shared with us by Doug Haig's lab. So we had purified from Drosophila embryos, that Eileen's lab, one of the beautiful experimental systems that they have in her lab, that they established in her lab, is you can cultivate Drosophila embryos at a huge scale, such that you can collect large amounts of precisely staged embryos and then fax purify, so using fluorescence activated cell sorting, different cell types. Okay. And so we had done muscles and neurons, and we had done this from different time points in embryogenesis. And we had done Capture-C, so looking at 3D contacts established by viewpoints. In total, we had 600 viewpoints. And these viewpoints, well, it was quite a large scale. And they were designed in promoters of genes that were either specifically expressed in muscle or in neurons, right? So tissue-specific genes.
So it was pre-informed, right? So you had your enhancers of interest, basically.
Yeah. So we designed the viewpoints in promoters and in enhancers, exactly. So enhancers that we knew were specifically active in muscle or in neurons, right? And then
so how did you determine which enhancers were active? Was it by transcribing a gene to different levels? Because a promoter activation is easy to measure, right? But how do you measure enhancer activity?
Right. So Alex Stark's lab created a wonderful resource that he published in Nature. Evgeny Kvon was the first author. They made thousands of Drosophila lines in which they cloned candidate regions, so in the genome upstream of a reporter gene, and then did thousands of RNA in situ hybridizations in order to find which expression patterns these tested genomic fragments could drive expression in. And so you can go online onto their website and simply, they've done a fantastic job at annotating these expression patterns, right? So you can say, okay, tell me all of the fragments that are expressed in muscle at this embryogenesis time point, and so we were just eyeballing this resource, and that's how we chose many of these candidate enhancers. There was also another resource published by a former colleague in Eileen's lab, James Reddington and David Garfield. They had done generated DNA-seq data sets in these back-sorted muscles and neurons at different time points in embryogenesis. So we also knew which accessible chromatin peaks were specifically accessible in either muscle or neurons at different time points. So we used all of these resources to design capture probes, as viewpoints. Okay. Yeah, exactly. And so what we found, this manuscript is on bioRxiv. We found that actually there are quite some examples of dynamic, let's say looping, I'm using quotation marks, events happening between tissue-specific viewpoints, that we see looping events only in the cells in which these elements are active at that specific time in embryogenesis. And so we see both kinds of contexts, some that are constitutive, right? They're always there, no matter which cell type you look at, no matter which time points, but they're also a substantial fraction that are dynamic, suggesting indeed that, context, the frequency of context can be modulated as a means to potentially enable, tissue-specific gene activation.
So the loop itself is not important, but it's just a contact, right? So the loop just is because the DNA is between the promoter and the enhancer. So the loop itself does not have a function, but it's the contact, right? So, for RNA, for rDNA, you know that you have this round of transcription that is going on in the loop so that when the polymerase finishes, it can start again. But the loop in those enhancer-promoter interactions does not have a function per se, right? It's just that there is the contact and the DNA is in between, and this is why you see a loop.
Yeah, so these loops occur typically between accessible chromatin peaks, right? And so there are probably contacts between promoters and regulatory elements, sites bound by transcription factors. In a lot of work that because this work was done with my former colleague Tim Pollux, who really did then most of the work after I left the lab to start my own lab. He really pushed this much further. He then tested, for example, some of these regions, these distant regions that a promoter contacted dynamically. He tested whether they are enhancers, and indeed they are. And they're enhancers, that were active at the time that you would expect or that you would predict based on when they were establishing this contact. And so, I think that many of these dynamic contacts represent an enhancer-promoter interactions.
Mm-hmm.
Yeah.
So when we go on the timeline and maybe moving away from CTCF, coming back to CTCF later, but there was also a paper where you talk about CP190. So was this connected to this study, or was this a follow-up study on the work you have been doing?
Yeah. So in Eileen's lab, I was fortunate to learn genomics, right, which was a set of skills that I acquired at that time in my career. And thereafter, to start my own lab, I decided to work. So after working on gene silencing, right, with Jörg, and gene activation with Eileen, I wanted to study gene insulation, which is the process by which you prevent promiscuous communication between a promoter and a regulatory element, right? So insulators are DNA elements that when you clone, when they're present in between a gene promoter and an enhancer, they're somehow magically able to prevent the communication between these elements, right? And
so it's like a bouncer in a discotheque.
It's like a what? A bouncer in a discotheque? Yeah, exactly. Exactly. The bouncer standing between you and the entrance of the discotheque. Exactly. So, right. So okay. When I started, it was unknown. So CTCF is the conserved insulator protein in flies and mammals. And Elfej Nora in Benoit Bruno's lab had recently published that CTCF in mammalian cells is required to form a large fraction of the boundary of TAD boundaries, right? So let me briefly say what TADs are. TADs are topologically associating domains, right? So when you look at how chromosomes are folded in 3 dimensions, you see that chromosomes consist of a succession of domains, okay, which you can think of as probabilistic 3D volumes that DNA within that domain tends to preferentially explore over time. And there are boundaries between these domains, and these boundaries are often occupied by CTCF in mammals. And if you deplete CTCF, you disrupt these boundaries. But in Drosophila, no one had really looked yet at what CTCF is doing. The Drosophila genome had been proposed to fold through completely other mechanisms than in mammals. For example, it had been hypothesized that there are no real structural proteins in Drosophila, that everything is just explained by transcription and transcription-related processes. And so when I started my lab, and actually I started making the CTCF mutants as a side project in Eileen's lab. And so together with Eileen, we published, the phenotype of these mutants, which is that surprisingly you remove whereas CTCF in mammalian cells, it's critical for a cell to survive. In flies, if you deplete CTCF, the flies make it to pupae, right? They're basically formed adults, but they just cannot hatch from the pupal case. And so that was unexpected because previously it had been, assumed that CTCF would at least be required for embryogenesis, which was not the case. So then in my lab, when I started my lab, we teamed up with Erez Lieberman Aiden, and I had the fantastic opportunity to go to his lab and learn Hi-C, because he had developed a low input protocol, which was crucial to be able, to be able to do Hi-C on dissected tissues from flies, right? As opposed to whole embryos or tissue culture cells, which is what had been done until then. And so we decided to look in the brain of these CTCF mutants because we saw that expression of CTCF in fly brains, that the fly brain is the most important organ that requires CTCF, meaning that you can make a fly that lacks CTCF everywhere in its body except for the brain, and it will die. And conversely, you can make a fly that expresses CTCF nowhere in the body except for the brain. Sorry, maybe I said that twice. And then it survives. So basically, expressing CTCF in the brain is sufficient to make a fly, to determine whether a fly will live or die. So that's why we performed Hi-C on dissected brains from these mutants, and we found that actually, just like in mammals, CTCF is required to form TAD boundaries in flies. The thing is that CTCF only binds to 10% of all TAD boundaries in flies. And so therefore it was only required to form about 10%. Right? So that showed us that that the mechanisms that are required to form TAD boundaries, they are conserved in flies, but the extent to which they contribute to forming all boundaries in the genome, this can vary widely, right? CTCF is required for 90% of TAD boundaries in mammals and only 10% in flies.
Which would also maybe hint to the fact that there must be something else, right?
Exactly. So there was clearly something else, and that's why in a follow-up study, we asked, well, what is this something else? And one obvious candidate that had been described in previous studies by other labs is a protein called CP190. And CP190 was reported to bind to all TAD boundaries in flies, pretty much. And this it's an interesting protein. It was discovered originally by Victor Corsi's lab in a genetic screen. So an unbiased genetic screen looking for a factor that is critical for insulator function. Okay, so for the ability of an insulator to block the communication between an enhancer and a promoter. And so we thought it was a good candidate. So we again generated CP190 mutants. By the way, we always use a genetic strategy that I developed when I was in Jörg's lab, by which we can make animals that completely lack both maternal and zygotic protein, which is important. It's basically these are animals that have never seen CP190 at any point in their life, right? And there we found that we lost many more TAD boundaries than in CTCF mutants. We lost a quarter, 25%, instead of 10%. So we lost CTCF dependent boundaries and in addition some other ones. And actually the results were pretty clear. Okay, so the 75% of TAD boundaries that persist in CP190 mutants, those are boundaries that are close to a transcribed promoter. So it means that in Drosophila, 3/4 of TAD boundaries are formed probably because of transcription. And Eileen's lab recently showed that indeed, if you mutate the promoter, you lose a TAD boundary, right? So indeed suggests that somehow transcription or recruitment of the transcription machinery, whatever, is sufficient to create a TAD boundary, a physical boundary. And a quarter, the remaining quarter of TAD boundaries in flies, they are distal, they're far away from a transcribed promoter, and they are formed by structural proteins like CTCF and other DNA binding proteins that exist in flies that are known to be insulator proteins. And what's funny is that all of these DNA binding proteins, they seem to recruit CP190 as a cofactor. And so whereas it's thought that in mammals, right, mammalian CTCF is sufficient to create a boundary, a TAD boundary, right, by acting as a barrier to chromosomal loop extruding cohesin. In Drosophila, CTCF needs to recruit CP190 to form a robust boundary. So somehow, fly CTCF and many other DNA binding proteins that can form TAD boundaries in flies, they all need to recruit this common cofactor. Okay, and so why are these results interesting? I argue that basically it showed that more mechanisms have evolved to form structural and regulatory boundaries than what was previously known from intensely studying this problem using mammalian cell culture, right? And so it's interesting to know that there are other mechanisms involved, and that, for example, the concept that a protein like CTCF can associate with a partner protein that makes it make a more robust boundary, right, is also, I think, interesting to know. Mm-hmm. Yeah.
So how does CP190 compare to CTCF structurally? Is it smaller, bigger? Does it bind to DNA? Does it only bind to CTCF? Do you know something about that?
Yeah, sure. So CP190, the name 190 means that it's 190 kilodaltons. So it's quite a big protein. Okay. It has 2 zinc fingers, and so it has been, debated about whether it can bind to DNA or not. From our findings and from those of many other labs, I think that CP190 cannot bind to DNA alone. It's really, for example, if we look at enriched motifs in DNA under CP190 chip peaks, the first motif we find is CTCF, because many CP190 binding sites are CTCF dependent. Yeah, CTCF is recruiting CP190 there. Then the next enriched motifs correspond to another DNA binding protein in Drosophila that's present at TED boundaries, suppressor of hairy wing. And then if you keep looking, then you find yet another DNA binding protein in Drosophila that binds to TED boundaries that's called BEEF32. Anyway, so basically all of these motifs that you find enriched on their CP190 peaks, they correspond to the sequences that are recognized by the DNA binding proteins that recruit CP190.
Okay.
So yeah, and then so one domain, protein domain that's present in CP190 that is 100% critical for its function is a BTB domain, which is so CP190 is a member of this family of transcription factors that's actually evolutionarily quite ancient. It's called the BTB zinc finger family of transcription factors. Mammals have about 40 members or so. And they are involved in a broad range of cellular functions, but it's thought that the BTB is an important homodimerization domain or heterodimerization domain. And so how exactly CP190 functions is $1 million question. And this I cannot say I cannot tell you anything apart from saying that the BTB is 100% critical.
And another question, how do insulators work? Do they just block like the scanning of other proteins on a linear DNA sequence, or can they also act in 3D and block the interaction by somehow, blocking the 3D interaction, or how is it thought that they work?
So that is the holy grail for us. I find it incredible that at this day and age, we still cannot answer the most fundamental question of how do you prevent a promoter from interacting with an enhancer, right? Just by inserting a DNA element. So this has been something that I've been trying to, my lab has been trying to answer. It is very difficult to say. So I think we have relatively strong evidence that insulators, at least in Drosophila, they do not function simply as roadblocks, that sit on DNA and that prevent an activating signal from scanning the DNA from an enhancer to a promoter. So this is, part of the evidence that I'm not describing because it's a little bit complicated to describe it in words. It's our next paper that we're preparing and that we're going to upload on bioRxiv in the coming months. But basically, we can reconstitute insulator function on a plasmid in a plasmid assay, and I think that the fact that we can do that is arguing against the model that insulators would simply act as a barrier. Yeah.
Is the degree of insulator function dependent on the linear distance from the insulator to the promoter?
No, it's not. No, well, at least not at the scales we've tested. At least not at the scales we've tested. Of course, studying insulation in a plasmid assay is artificial, obviously, right? I mean, what we see, but nevertheless, the properties that we are able to dissect using our plasmid assay, they make a lot of biological sense. And so, on the one hand, you could argue that, okay, this is very artificial. We cannot conclude anything. But at the same time, none of the currently considered models, of how insulators function can explain our findings. So in a way, even if this were completely artificial, I think it's still very intriguing. How are these things working, right? So yeah, that's the $1 million question. How do insulators function? Yeah.
We've now come a little bit away from your work and dove into like the more meta work, but there is one piece of science that we have not discussed from the timeline or from your timeline, and this is a paper that was just published a few weeks ago in August 2023, and this was published in Cell, and there you exactly talk about those distant TED pairs, megabases apart, and how they can interact to form metadomains. Maybe we can close the science that has been done in your lab by talking about this, and you just talking about that for a bit.
Sure. So this all started because we had done, we had looked at chromosome folding in brains of CTCF mutants in this first study. And actually another phenomenon that we had observed but had not described in that paper because it was unrelated to TAD boundaries is that we had seen very, very distant associations between distant TADs, okay, that were millions of base pairs apart. And so in this other study that just came out and with which we teamed up with Alexander Jankowski, who is a computational biologist in Warsaw, and the lab of Mike Levine in Princeton, who actually had better maps than we had. He had micro C maps, which is higher resolution than what we had, high C. Basically, we found that this was a previously unrecognized level of genome folding in which very specific pairs of TADs, in the most extreme cases, on one end of the chromosome coming together with the other end of the chromosome, right? That they're able to come together. They come together because they are tethered. They are physically tethered by punctual interactions between sites bound by transcription factors in one TAD and in the other, okay? And so if we cut these tethers, for example, by making precise CRISPR-Cas9 deletions of one of the, what we call anchor sites, or by mutating the transcription factors that bind to these anchors, in both cases, this is sufficient to completely blow apart these TADs, okay? So these metadomains, these associations between distant TADs, they are tethered by these punctual interactions within them. Interestingly, these punctual interactions involve promoters of a subset of neural genes that are involved in axon guidance and signaling. For example, we have glutamate receptors, serotonin receptors, and acetylcholine receptor genes looping. And we have genes that encode proteins involved in axon guidance that are looping. And so these gene promoters are looping to intergenic elements. And by perturbing these physical associations, we found several, so in total 4 examples of neural genes that when we disrupt the pairing, their transcription went down, okay? And so these very distant inter-TAD associations had been previously noticed in mammalian contact maps in mammalian cells, but it had been very difficult to assess whether they were functional or not, because in mammals, these associations between distant TADs are much more complex. You have many more TADs coming together, right? And so if you perturb one, it was very hard to see an effect on the gene in one of the distant partners, right? But in Drosophila, because these distant TAD interactions are so specific, frequently we have 2 pairs of TADs coming together, right? We were able really to make precise perturbations and disrupt, okay, these associations. And in many cases, this affected the transcription of a gene that was more than 1 million base pairs away. And so ultimately, these represent the longest range regulatory associations that have ever been described in any genome, which I find quite funny because typically Drosophila is considered to be a compact genome, and yet that's where we have found, the largest range interactions so far.
Maybe that's the reason for that, right? Because it's also compact, it's closer together. Yeah,
that could be, yeah.
So this already leads us to the question: what are your plans for the next 5 years? Right? Is that exactly what you want to do in the next 5 years or for the next grant?
Right. So we started the lab working on TAD boundaries. It kind of enlarged to studying inter-TAD associations. Now I can explain our research as ultimately trying to understand how the specific communication between promoters and regulatory elements is organized in 3 dimensions. So for our research on TAD boundaries, the holy grail that we were discussing before is to understand how boundaries can prevent the communication between an enhancer and a promoter. Current models don't explain our findings. We're trying to come up with new models to explain this. For inter-TAD associations, what I find one of the most remarkable features of what we found, in my opinion, is the specificity, right, with which these DNA elements that are millions of base pairs apart, how do they find each other, right? It's incredible. Is it encoded in the DNA, or is it also that where these sequences are in 3 dimensions in the nucleus, whether that is another important layer to bring them together? So we have projects that are going to address these kind of questions. And finally, the question is, why does this specifically happen for neuronal genes and not for other, genes of different cell types? What is it about neural genes that make them special? And so this is another holy grail, very difficult question to address, but that we have to think about.
This is true for only Drosophila, right?
No, in mammals as well. So if, in mammalian cells, the most striking long-range 3D contacts that have been described occur in mammalian neurons.
Okay.
And actually they occur between olfactory sensory receptors. So again, genes involved in signaling, right? And so it seems like animal neurons, we want to now explore how broader a concept this is, apart from just knowing that it exists in flies and in mammals. What about other species in the tree of life? How many of them show very long-range contacts between neural gene promoters and very distant elements?
Okay.
Yeah.
So for the last now 44 minutes, we have taken a journey through your scientific career. Did we miss something important, or would you like to add something?
No. I'm surprised we talked this long, but I think we really went into great depth. So no, I don't think we missed anything.
So to finish, could you give us a brief summary of your most important finding?
Oh dear. So yeah, I don't know. I mean, it's hard for me to rank them. I would just say maybe more broadly, the fact of approaching questions that are intensely studied using one model, and the fact that we use Drosophila, right? I think getting this kind of evolutionary perspective of what is possible, right? What different ways has life come up with to deal with similar challenges? I think it's an important approach and an important insight to kind of broaden the view of what is possible and what is necessary. Yeah.
So thank you, Maria, for your time and for being on the show.
Thank you very much, Stefan. It was a pleasure.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.